whisper some nice science while you get lulled to sleep. Today, we're going to talk about molecular evidence of anterior-posterior patterning in adult echinoderms. This was published in Nature, and it's by L. Formery, B. Peluso, I. Conley, J. Malnick, J. R. Thompson, M. Patel, K. R. Ulinger, D. S. Roxar, D. R. Rank, and C. J. Lau. Abstract: the origin of the pentaradial body plan of echinoderms from a bilateral ancestor is one of the most enduring zoological puzzles. Because echinoderms are defined by morphological novelty, even the most basic axial comparisons with their bilaterian relatives are problematic. To revisit this classical question, we use conserved anterior-posterior axial molecular markers to determine whether the highly derived adult body plan of echinoderms masks underlying patterning similarities with other deuterostomes. We investigated the expression of a suite of conserved transcription factors with well-established roles in the establishment of anterior-posterior polarity in deuterostomes and other bilaterians using RNA tomography and in situ hybridization in the sea star, Bacteria miniata. The relative spatial expression of these markers in P. miniata and ambulacral ectoderms shows similarity with other deuterostomes, with the midline of each ray representing the most anterior territory and the most lateral parts exhibiting a more posterior identity. Strikingly, there is no ectodermal territory in the sea star that expresses the characteristic bilaterian truncogenetic patterning program. This finding suggests that from the perspective of ectoderm patterning, echinoderms are mostly head-like animals, and provides a developmental rationale for the reevaluation of the events that led to the evolution of the derived adult body plan of echinoderms. Echinoderms, defined by their calcitic endoskeleton unique water vascular system, and perhaps most strikingly by their pentaradial body plan, are among the most enigmatic animal phyla. Because echinoderms are phylogenetically nested within the deuterostomes, echinoderms, hemichordates, and chordates, their pentaradial organization was evidently derived from a bilateral ancestor. Yet, despite a rich fossil record, comparative morphological studies have come to conflicting conclusions about the axial transformations that led to the pentametry from the ancestral bilaterian state. Among bilaterians, the deployment of the gene regulatory network that specifies ectoderm anterior-posterior AP polarity is highly conserved and represents a suite of characters that is often more conserved than the body plans they regulate. Figure 1a. The deployment of this AP gene regulatory network could therefore provide an alternative way to test axial homology in cases such as echinoderms, in which morphological characters are too divergent to reconstruct ancestral states. Detailed comparisons between chordate and hemichordate axial patterning have established the exquisite conservation of the deuterostome ectodermal AP patterning program, which provides a phylogenetically robust framework for addressing the evolution of echinoderm axial properties. We consider two contrasting scenarios. First, the ancestral deuterostome AP patterning network could have been dismantled and reassembled into new conformations during the radical body plan modifications along the echinoderm stem lineage. In this scenario, expression of transcription factors in derived morphological structures without extensive conservation of relative spatial expression would imply co-option into new developmental roles. Alternatively, conservation of spatially coordinated expression of this network during the elaboration of the echinoderm adult body plan would provide a molecular basis for testing axial homology with bilaterians and establish regional homologies masked by divergent anatomies. Under the second scenario, 4 main proposals were made to relate the echinoderm body plan to other bilaterians that explicitly consider axial homology, figure 1c. The bifurcation and the circularization hypotheses can be ruled out since they require a unique molecular identity for each of the 5 echinoderm rays that is inconsistent with molecular data. In the duplication hypothesis, each of the 5 echinoderm rays is a copy of the ancestral AP axis joined anteriorly in the disc, and in the stacking hypothesis, the oral-aboral axis of the double echinoderms is homologous to the ancestral AP axis, although broad bilaterian comparisons of AP axis patterning are typically based on ectoderm expression domains. The stacking hypothesis was proposed largely on the basis of nested posterior Hox gene expression in the posterior mesoderm of the bilateral larval stage in holothurids, sea cucumbers, sirenoids, sea lilies, and echinoids, sea urchins, although initially restricted to coelomic compartments. Some iterations of the stacking hypothesis have also included the oral ectoderm. Ectoderm has the most anterior compartment. However, recent analyses suggest that the anterior identity of the oral ectoderm should be considered independent of the mesoderm derivatives. Here we tested these classical hypotheses by examining gene expression in the asteroid C-star Patricia miniata extended data figure 1a-b using a statistical approach based on spatial transcriptomics and in situ hybridizations. Our data provide evidence for unexpected conservation of the ancestral AP ectodermal patterning network during ambigularcrum development in juveniles. Importantly, our results do not fit with any of the established hypotheses, and instead suggest a new ambulacral anterior model to relate echinoderm parent pentaradial symmetry to the ancestral bilateral symmetry. RNA tomography in adult Pimeniata. The manifestation of pentaradial symmetry in asteroids is simpler than in other echinoderm classes because it follows a planar organization with the rays extending around the oral-aboral axis through easily distinguishable alternating ambulacral territories where the grooves burying the 2 feet are located, and interradial territories, figure 2a. This makes asteroids well suited for investigating potential axial homologies between the pentaradial echinoderm body plan and closely related bilateral deuterosomes using the molecular anatomy coordinates offered by a conserved ectodermal AP patterning network. The adult body plan of asteroids is defined by the endoskeleton, which underlies the body wall in the aboral and interradial areas, the digestive tract and pyloric caecae, the main body muscles, the water vascular systems along each ambulacrum, and the central nervous system that includes 5 radial nerve cords, RNC, and the circumoral nerve ring, CNR. We used microcomputed tomography, micro-CT, to show the exact spatial arrangement of the principal anatomical features in P. miniata juveniles along each organizational plane of the juvenile arms, figure 2a, extended data figure 1c, d, supplementary figure 1, and supplementary video. Within this anatomical framework, we began investigating the transcriptional landscape of P. miniata along its body axis with an unbiased spatial transcriptomics approach using RNA tomography. We cryosectioned 3 arms from P. miniata juveniles at a stage that matched the micro-CT scan specimens along 3 different dimensions, from the proximal to the distal part of the arm, P to D, from the oral to the aboral side, OA, and from the left to the right side, hereafter referred as mediolateral, ML. Figure 2b, extended data figure 1c, supplementary video, and supplementary figure 2ac. Sections were barcoded and pooled for single molecule real-time sequencing with PacBio IsoSeq supplementary figures 2D, E, and 3AD, and supplementary tables 1 through 4, yielding a 3-dimensional atlas of 25,794 gene expression profiles along the P to D, O to A, and ML dimensions of P. miniata arms. Gene clustering based on the similarity of RNA tomography expression profiles highlights 7 principal patterns of gene expression, figure 2c. We confirm that transcriptional landscapes determined by RNA tomography was consistent with anatomy of the animal considering the principal component analysis, PCA, and Spearman correlations between sections along each dimension and analyzing the expression profile of marker genes known to be expressed in specific tissues. Extended data figure 1E through G. To aid in this analysis, we generated a new P-Miniata genome assembly, supplementary figure 2E and 3D E, and supplementary tables 4 and 5 to consider possible molecular anatomical homologies across deuterosomes. We identified 36 conserved molecular markers in the RNA tomography dataset that define specific ectoderm territories along the AP axis in hemichordates and chordates, figure 1A and supplementary figure 4, and retrieve their expression profiles. These marker genes include transcription factors FESF636 and KX2.1, FOXG LHX2/9, OTP, DUMBX, TLX, EMX, BARH1, PAX6, IRX, TBX, OTX, LHX1 and 5, NGRAIL, GBX, PAX258, HOX1, HOX2, HOX3, HOX4, HOX5, HOX7, HOX8, HOX9/10, and HOX11/13B. Members of the Wnt signaling pathways, SFRP1/5, SFRP3/4, FZD5/A, and Wnt3, and the ligand hedgehog. 4 more transcription factor markers, RX, DLX, HOX11/13A, and HOX11/13Z, were excluded from the computational analysis because of low expression levels, supplementary figure 3F. HOX6 was absent in our HOX cluster assembly, supplementary figure 5, as reported previously in the closely related species P. pectinifera, and in the crown of thorns sea star, Acanthaster plantae, consistent with its loss in valvidate asteroids. The duplication and stacking hypothesis would be supported by staggered expression of these A to B patterning markers along the PD and OA dimensions respectively. We ranked the AB patterning markers from the anterior to posterior using MA chordate Saccharomyces Kowalewski as a template, as it is the most closely related bilateral species with a comprehensive expression pattern dataset for these markers, supplementary figure 6. We then tested the Spearman correlation between the ranking of the genes and their position along the PD and OA dimensions. In both cases, we found moderate, but not statistically significant correlations. P equals 0.25, B equals 0.216, and B equals 0.27. P equals 0.13 respectively. Figure 2E, organizing the AB patterning genes into 7 groups on the basis of their expression profiles, supplementary figure 6B. We found that the moderate correlation with the O to A dimensions was mostly explained by the HOX genes along figure 2F. This was expected because the stacking hypothesis was primarily informed by the sequential expression of the HOX genes in mesoderm derivatives. Unexpectedly, we found a much stronger correlation, P equals 0.62, P equals 1.4 times 10 to the minus 4, between gene order and the ML axis, figure 2E. The most anterior genes appear to be largely expressed close to the midline of the arm, whereas more posterior genes were expressed more laterally on either side of the midline. To confirm that the observed correlations were robust and not the result of the sensitivity to the AB patterning gene ranking assigned from the in situ hybridization expression data, we simultaneously shuffled the gene ranking within each of the 7. Each of the 7 groups and probabilistically sampled the position for each gene using the expression z-score as a law of probability in 10 to 6 replications. For all the simulated replications, the distribution of ML correlations was significantly superior to the PD and OA correlations with a 2-sided Wilcoxon rank test. p-value inferior to 10 to the minus 4 in both cases. Figure 2e. This suggested that neither the duplication nor the stacking hypotheses accurately describe the deployment of A to B, A-B-related patterning genes in P miniata, and that the most underlying patterning logic of the pentaradial plan is not explained by existing models. ML deployment of AP patterning genes. Although RNA tomography provides coarse average axial positional information, these patterns cannot be directly linked to germ layers or anatomical structures. To investigate axial patterning in more detail, we examine the expression patterns of the 36 markers using in situ hybridization chain reactions, HCRs, supplementary figure 7, on post-metamorphic juveniles. This representative stage follows the reabsorption of larval structures into the rudiment during metamorphosis. The elaboration of the pentaradial symmetry is initiated earlier within the larval mesoderm, but the mechanistic basis of this process is probably distinct from those give their regional identity to the final axes of the pentaradial body plan, expression of classic ectoderm AP patterning genes, such as 636, NK2.1, DMBX, PAC6, and OTX, was initiated at low levels in the presumptive adult tissue at the onset of metamorphosis, but only reached robust levels of expression, which were relatable to ambulacral polarity in post-metamorphic juveniles. When the definitive adult body plan is elaborated, Figure 3 and supplementary Figure 8, we focused on early juvenile stages as a readout of early establishment of axial patterning. As in the adult, the juvenile body plan is organized into ambulacral territories on the oral side and interradial territories at the edge of the ambulacra and extending around to envelope the entire aporal side. The ambulacral ectoderm is divided into main regions: the medial ambulacral ectoderm comprising the RNCs and the CNR, and more laterally the epidermis covering the podia. Figure 3c, D. We analyzed the expression of 4 groups of patterning genes expressing the AP patterning in both hemichordates and chordates. The first group has strong anterior ectodermal localization in the proboscis of S. Kowalewski and in the forebrain of vertebrates, Figure 1a, B. In P. medianata, we found similarly overlapping patterns of regional expression, mostly restricted to the developing CNR, the RNCs, and in the case of 636 and hedgehog, in repeated domains where lateral nerves from the RNCs. Connect with each pair of secondary podia, Figure 3e to L, and supplementary Figure 9a to E. This region corresponded to the most medial parts of the ambulacral ectoderm, Figure 3d. These findings are inconsistent with the duplication hypothesis, which predicts staggered expression of anterior to posterior markers along each RNC. The next group overlaps with the previous anterior class genes in S. Kowalewski, but with a more caudal distribution in the posterior proboscis and into the collar. In chordates, these genes are primarily expressed either in the forebrain or in the midbrain, Figure 1a, B. In P. medianata, expression of this group of genes overlaps in the most medial territory with the most anterior class genes, but with expanded lateral domains on either side of the RNCs, Figure 3d, M through S, and supplementary Figure 9, F through H, into the epidermis covering the podia. FES-F and DBX were only expressed in a few cells in the ambulacral ectoderm. Supplementary Figure 10a through C. Only PAC6 was not expressed in the medial ambulacral ectoderm and was restricted to the podia epidermis, as previously reported in other echinoderm species, Figure 3c. Thus, the lateral ambulacral ectoderm appears to have a more posterior molecular identity, similar to that of the hemichordates in the hindbrain into the midbrain/hindbrain boundary. Wnt3 is expressed at the far posterior end of the AP axis in both phyla. In P. meonata, we found that these 4 genes are expressed at the boundary between the ambulacral ectoderm and the interradial territory, figure 3D, U through AB, and supplementary figure 9, I through L. GBX and Wnt3 were expressed in the outer part of the ambulacral ectoderm, establishing a mutually exclusive boundary with more anterior genes like 6-3/6, figure 3U through X. HOX1 and PAX258 were expressed more laterally compared to GBX and Wnt3. HOX1 outlined the entire ambulacral area, whereas PAX258 had a more complex expression pattern and was primarily expressed between the ambulacra, figure 3Y through AB. We suggest that in P. meonata these genes marked the outer limit of the anterior compartment. Finally, HOX genes are expressed in hemichordates and chordates posterior to the collar/trunk and midbrain/hindbrain boundary, respectively, and are involved in trunk patterning, figure 1AB. In P. meonata, only HOX1 was detected in the ectoderm. HOX3, HOX5, HOX8, and HOX9/10 were expressed in mesoderm derivatives, figure 3AC to AF. HOX4 expression was barely above the detection threshold, but was found in the pharynx, figure 3AG, whereas HOX7, HOX11/13A, and HOX11/13B were expressed in the developing intestinal tract, figure 3AH through AJ. These observations are in line with previous reports that HOX expression in echinoderms is largely restricted to internal germ layers and consistent with the HOX-driven O through A correlation observed in our RNA tomography dataset. In hemichordates and other bilaterians, HOX gene domains are intercalated during trunk development between the anterior domains and the posterior end of the animal, which expresses posteriorizing factors such as Wnt3. We propose that there is no ectoderm equivalent to a trunk region in P. meonata because Wnt3 is expressed at the edge of the ambulacral region, and because HOX1 is the only HOX gene expressed in the ectoderm. Therefore, the deployment of A through B patterning system in P. meonata seems to be limited to the ambulacral region and its boundary. We also found that in P. meonata, some ancestral deuterosome AP patterning genes did not exhibit conserved relative expression compared to the hemichordates and vertebrates, either because they were not detected, not expressed in the ectoderm, or exhibited different relative spatial arrangements. Supplementary figure 10D through I. Some well-established boundaries in hemichordates and vertebrates, like the abutting OTX and GBX domains, were also not observed in P. meonata. This presumably reflects the plasticity of the A to B patterning system and its adaptation to the radically different pentaradial body plan. Despite these discrepancies, germ layer-specific expression patterns corroborated that the M to L dimension revealed by RNA tomography was the main and unexpected driver of ectoderm AP patterning logic of the pentaradial body plan of P. meonata. If the ancestral deuterosome AP patterning system is used to remodel the C-star body plan and metamorphosis, are other correlated bilaterian axial patterning systems contributing to the patterning of the adult body plan as well. In bilaterians, the dorsoventral DV axis is specified transiently through embryogenesis by conserved BMP2/4 Gordon gradients. Later, the neuroectoderm and mesoderm derivatives are patterned along the DV axis via the deployment of transcription factors such as PAX3/7, MSX, and TBX2/3 in response to BMP signaling. Extended data figure 2A to investigate whether the BMP coordinate axis is involved in the formation of the adult body plan in P. meonata. We first looked at expression of genes involved in DV axis specification through metamorphosis and in post-metamorphic juveniles. Extended data figure 2B through S and supplementary figure 4. Although coordinate was not detected by in situ hybrid hybridization, we found that BMP24, BMP1, and ADMP1 were consistently expressed in the distal part of the developing tube-feet mesoderm, starting early during metamorphosis and into the juvenile. Extended data figure 2C through S. In addition, we looked at known target genes of BMP signaling, extended data figure 2B through T, V, and supplementary figure 4. Although PAX37 is lost in echinoderms, TBX23 and MSX were co-expressed with BMP24 and 2V, and TBX23 was also expressed in the overlying 2V ectoderm, suggesting a similar relationship to BMP signaling to that previously demonstrated in vertebrates. Extended data figure 2T through V. These expression data are consistent with previous studies from a direct developing echinoid and support a role for BMP signaling in tube-feet development, more equivalent to the later role of BMP in dorsal midline patterning in hemichordates and vertebrates. However, our results do not indicate BMP chordin signaling as defining an anatomical axis in the pentaradial body plan of P. miniata. Echinoderm rays have also been proposed to be homologous to bilateran appendages. In principle, this scenario could be compatible with our expression data, with the rays being appendages of anterior territory. To assess this possibility, we consider conserved markers for PD appendage development, which involves polarized expression of mice PBX, DLX, and SP89 in vertebrates, arthropods, and cephalopods. Extended data figure 2WX and supplementary figure 4. Although we found the expression of PBX and particularly SP89 to be compatible with this idea, DLX was not detected and mice had an inconsistent expression domain. Extended data figure 2X through alpha. The extension of the echinoderm ambulacra probably involves substantial development novelty. The RNA tomography dataset will be an invaluable resource for hypothesis generation identification of candidate genes to form the basis of a more in-depth development investigations of this process. Evolution of axial properties in echinoderms. The organizational modifications to the ancestral bilateral deuterosome body plan during early echinoderm evolution are so profound that even basic axial comparison with the other deuterosome taxa have been problematic at the morphological level. Here we investigated this fundamental question by spatially mapping the deployment of our ancestral bilateran ectodermal AB patterning system in the pentaradial body plan of P. miniata. Because this patterning system is largely conserved between hemichordates and chordates, we can confidently reconstruct its ancestry in early deuterosomes and at the base of ambulacranians. We found that much of the ancestral interior patterning network is spatially deployed in a manner incompatible with previously proposed hypotheses of echinoderm axial homologies. Rather, expression patterns map onto a new coordinate system, which we call the ambulacral anterior model of echinoderm body plan evolution. Figure 4a-b. In this model, the midline of each ambulacrum expresses the most anterior bilateran molecular identity, equivalent to the forebrain and proboscis in vertebrates and hemichordates, respectively. The midlateral regions of either side of the nerve cords, including the ectoderm wrapping podia, share patterning similarities with mercadal ectodermal territories of hemichordates and chordates, down to the collar and midbrain, respectively. Finally, the boundary at the edge of ambulacral ectoderm displays the most posterior molecular profile, corresponding to the collar trunk boundary of hemichordates and the midbrain hindbrain boundary of vertebrates. According to our ambulacral anterior model, echinoderms are the first example of bilaterians, in which the anterior identity is located at the center of a sheet of tissue rather than being located at the extremity. The anatomical outputs of this anterior domain, however, share similarities across all deuterosome phyla, including echinoderms, in that they include neural condensations and extensive array of sensory structures. absent in the sea star except for OX1, suggesting a loss of the ancestral ectodermal trunk regulatory program, which is consistent with previous observations in the echinoid Paranella japonica, yet OX genes are expressed in the mesoderm and endoderm, displaying a marked uncoupling of germ layer AP patterning as recognized previously. The mesoderm and endoderm trunk programs are wrapped in an ectoderm with two clear territories, including one, an ambulacral anterior-like domain that is the main focus of our work, and unexpectedly two, an extensive interradial domain that extends around the aporal side of the animal and displays uncertain axial identity without any detectable readout of the ancestral AP patterning program, as previously observed in echinoids. The uncoupling of an ectoderm head and trunk programs is not unique to P. miniata and has been demonstrated in both larval echinoderms and hemichordates, and more recently in protozoan larvae, suggesting that these regulatory programs can be uncoupled over macroevolutionary timeframes. Previous studies propose that the deployment of transcription factors to pattern adult echinoderm body plans could be the result of co-option, meaning recruitment of an existing gene or regulatory module or duplication thereof to fulfill a new developmental role. Here, using a much larger set of genes, we demonstrate that the patterning of the ambulacral ectoderm in P. miniata involves a collection of interacting modules that define distinct regions of the AP axis of bilateral animals rather than single regulatory program. Co-option of this network would require redeployment of these interacting modules into a new role in stem echinoderms. Thus, though we acknowledge that the co-option remains a possible scenario, we propose that the observed pattern in extant asteroid ambulacra is a result of modification of ancestral deuterosome axial program during echinoderm evolution. Documented cases of co-option, such as the deployment of posterior OX genes during limb patterning, involve the reuse of an existing regulatory network to pattern a new structure without affecting the ancestral role of the system. To our knowledge, there is no example of co-option that involves dismantling of the ancestral patterning networks following their recruitment into the development of a new structure. A co-option scenario in echinoderms would imply that the ancestral function of the AP patterning network was lost in the body plan after co-option into an evolving ambulacral territory, which in our opinion is less parsimonious than proposing descent with modification of an ancestral AP axis. Interestingly, a route to resolve this issue might come from some interpretations of homolozoan and helicoplacoid echinoderms that propose these groups to be transitional forms between bilateral and pentaradial symmetry. Reinterpreting these fossils in light of a new patterning datasets could allow us to discriminate between co-option with loss of the ancestral axial registry or descent with modification. And the reorganization of ancestral AP axis documented here occurred during early stem evolution of echinoderms as a defining regulatory feature of ground group echinoderms, or during later diversification of asteroids. Our model, inferred from analysis of asteroid data, is consistent with published findings from two other main echinoderm clades, cyrenoids and echinoids. In cyrenoids, the expression of 636, OTX, and PAC6 has been reported in the ambulacral ectoderm. In echinoids, AP patterning-related genes have been surveyed in various species, including Strongylocentrotus purpureus and Heliosideris erythrogramma, and most expensively in P. japonica, in which the anterior identity of the ambulacral ectoderm was already reported. In both cyrenoids and echinoids, most of the anterior patterning-related genes studied showed expression patterns compatible with the ambulacral anterior model. Furthermore, OX gene expression has been described in echinoids, holothuroids, and cyrenoids, and is largely congruent with the expression patterns that we observed in P. miniata. This suggests that on the basis of limited available comparative data, the ambulacral anterior model could be a general neurodevelopment feature of all extant echinoderm classes, and that our observations reflect early regulatory changes which occurred during stem echinoderm evolution. Figure 4c. Further comprehensive comparative analysis will be required to test this hypothesis. The evolution of body plan variations across echinoderm classes has proven challenging to reconstruct on the basis of morphological features alone. The ambulacral anterior model offers a powerful tool to establish robust regional homologies between echinoderm classes. Most importantly, the new axial paradigm established here can be integrated with the exquisite fossil record of the phylum to reinvestigate key morphological transformations in light of regulatory changes. Online content, any methods, additional references, nature portfolio reporting, summaries, source data, extended data, supplementary information, acknowledgements, peer review information, details of author contributions and competing interests, and statement of data and code availability are available online. Thanks. Thanks so much for listening.